Hello and welcome to this video on how the immune system recognizes things it should attack. These clues are known as antigens. Without antigens, you'd not be able to differentiate between your cells when they behave, when they misbehave, and foreign cells like bacteria. Antigens are the cornerstone to how vaccines, immunity, and immunotherapies work. These are the site to which antibodies bind. These can be found on the outside of the cell, virus, bacterium, parasite, and so on. Antigens are special because they are very unique. Think of them as similar to a barcode for every possible feature that could be identified. These markers are made from proteins, amino acid chains, polysaccharides, but not lipids or nucleic acids when in isolation. When you find lipids and nucleic acids with protein, they do become antigens. Your antibodies are specific to antigens. While not exactly a lock and key model, it is a good analogy, with one antibody going to one antigen. The right antigens can be seen as either self or your own cells. This leads to the non-self antigens as an alternative. A simple but effective distinction. The immune system is developed in such a way that it will let slide those cells it recognizes as being its own or your own cells. Those that are not self cells or not self antigens must be targeted and destroyed. It is also a useful phenomena in quick antigen testing. Such specific antigens can be identified by equally specific antibodies. Testing this way is reliable, accurate, easy, and often relatively cheap. Antigens are needed and are the basis for an immune response. To understand what happens with an antigen, we need to look at it in the context of an antibody. The antibody has a specific set of sequences at the top end of the Y. These are highly variable, and so the body can produce a vast number in different configurations. These can match to one specific antigen based on how it's configured. The area that very highly variable area binds to is called the paratrope. The area of the antigen that interacts with the antibody is known as the epitope. The degree to which these two match together and create a stable connection is called avidity. The importance of this is that although it's not a perfect lock and key model between the two, it's close, but it also allows for a degree of flexibility in just what the antibody can bind to. Closely related antigens could have multiple antibodies binding to them. In order to get the antibodies that will bind to these antigen sites, you need to begin producing the antibodies, but you need to also go several steps back before that can occur in a large amount. The first thing that occurs is that you have to have a macrophage. These are large cells that can consume other foreign material or infectious agents, such as bacteria or viruses. These macrophages can go along and eat the foreign matter, and then from this it can begin breaking it down. It can break it down and then find the antigens and give these to something called an antigen presenting cell. These take these to dendritic cells, and the dendritic cells act as a central hub which coordinates the target that the immune system is looking for. These are often found in your lymph nodes. From these dendritic cells, the other cells get their instructions on where to go and what to do. It does this by presenting those small bits of the antigen on the surface on these big indicators called major histone complexes. There are two types. These are called MHC1 and MHC2, MHC being major histone complex. MHC2 is found on the top of these antigen-presenting cells, and that's why when the immune cells get the signal from it, they know that this is their new target. When they find something similar on an MHC1 presenting surface, they know that that cell has been affected by whatever the antigen has come from. This tells the immune cell to attack that particular cell. If in the case of a viral infection, it destroys a cell infected by the virus. If it's an MHC2 complex, with the virus on the top of it, it knows that it's now looking for that bit of virus. In order to make cells more obvious to the rest of the immune system if they are infected, or in order to find things that are the source of the infection, you need to have a group of cells called B cells. These B cells are what produce the antibodies from a specific subset called plasma cells. These plasma cells also get their instructions based on the MHC complex. This allows them to know what they're trying to generate antibodies for, or perhaps more accurately it would be said that the body selects B cells based on which antigen it's looking for, as the B cells are generated at a random set, and the random set is then shuffled to try and find the right B cell that will produce the right antibody. Once you have found a B cell or a B plasma cell that can produce the right antibody, some of these are changed into memory cells which store the particular antibody for use later if required. This allows the immune system to respond better. Selecting for the right plasma B cell takes time, and it's why it can take several weeks for your body to start making antibodies to something foreign, or more accurately to a non-self antigen, and in some cases self-antigens for cells that have turned rogue, such as tumours. Once the problem that is presenting the antigen has been dealt with, the immune system starts to decrease the number of antibodies it produces, and it either removes the unneeded B plasma cells or converts them into those memory B cells for use later if required. This is one reason why, once you have measles as a child, your body generally handles it quite effectively and it doesn't come back a second time unless your immune system is somehow temporarily compromised. This is an event known as shingles. Although we won't go into a lot of detail here, it's worth knowing that antibodies is a broad description of a number of things. There are three important ones. These are known as immunoglobulin M or IgM, immunoglobulin A, IgA, or immunoglobulin G, IgG. IgM is the first antibody produced to deal with an antigen. The second, IgA, can be found in your saliva and other bodily secretions. IgG is the type found in all bodily fluids. Your immune system generally transitions from making IgM to making IgG. We've already mentioned that in order to get your antibodies, you need to find the right B cell. B cells are made in your bone marrow, same place as your erythrocytes. In order to get the right B cell, you need to make a vast number of them. In fact, hundreds of trillions of B cells. Each of these has the ability to make an antibody that's specific and different from the other B plasma cells. It must, however, also remove any B plasma cells that create antibodies that detect the self as being foreign. When this doesn't happen, you have antibodies that detect the self as foreign, and it leads to either autoimmune diseases and/or allergies. What happens when you have this is that the B cells create an antibody that binds to the antigen, and this signals for apoptosis of the cells. This is obviously not something desirable in your body, as you want to try and keep cells as much as possible and not have your immune system slowly destroy your own body. Selection of the right antibody occurs by having the B cells attached to the right antigen. This often occurs by things like the lymph organs, nodes, and so on, where they can find a mass amount of the antigen present from all the immune cells, bringing it back there for things like those dendritic cells. Once the B plasma cells know what they're meant to be doing and have the right instructions set, they go back to the bone marrow, and that's where they start pumping out their particular antibody. These B plasma cells are only able to act for a finite period of time. Much like erythrocytes, which only survive for 120 days because they have one mission, which is to transport oxygen, your B plasma cells are similarly oriented. They are there only to produce as many antibodies in a finite period of time as possible. These antibodies are in and of themselves quite interesting. They are made up of 4 parts that are relatively identical: 2 heavy chains and 2 light chains. At the top of the heavy chain is where you find the highly variable region. This is sometimes called the FC region. The FC region is what's going to bind to the antigen site. The rest of it is called the constant region, and it's relatively stable. This means that you can have a large number of B cells that only need to make a small tweak to what they're producing to have a vast ability to adapt to many different antigens and then be able to select from a vast array of B cells. The antibodies themselves, once they have bound to this antigen site, then help the other immune cells identify the foreign antigen. Coating something like, say, a bacterium with a whole bunch of flags in the form of these antibodies then allows for the immune cells to more readily and easily recognize what they're looking for and take action. It's kind of like a game of Marco Polo. Without the antibodies floating around, you would have to be looking for everybody else in the game without anybody calling back when you call out Marco. The antibodies, to an extent, act as someone calling out Polo in response to your call. You can then move towards them better, and you're not rummaging in the dark blindfolded, trying to eventually stumble across what you're after, or in this case, what the immune cells are after. A common antigen that is particularly useful, especially when you're in a hospital, is that of blood typing. On the top of every blood cell, barring the O negative, you have a particular marker. You have both the blood type and the rhesus type. If you have the antigen for a rhesus, then you are rhesus positive. If you have the antigen for A, B, or O, you are the blood type. In terms of foreign pathogens such as bacteria, mannose is a good example. It's largely not used in human cells, but can be found in the bacterial wall. This means that if you have antibodies particular to mannose, then you are able to more readily target that without concern for affecting the rest of your body. Whilst this has been a very broad summary of what antigens are and how they are used by the immune system, it should give you a strong idea as to what they are, where they are, and how they work. In the future, there will be more videos, particularly focusing on both immunoglobulins and particularly how the antigen, antibody, and immune system as a whole makes use of this information to provide you with long-lasting protection, for example, after a vaccine is administered. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.